Welcome, first year students. Today we will start with chapter 4 of Guyton, which is about the transport of substances through cell membrane. First you should know the significance of these transport mechanisms across the membrane. That why we have to look into the details of all these transport mechanisms, because my dear, membrane transport, it is a basic physiological process, and I hope that so far you know that there are certain differences between the composition of intracellular fluid and extracellular fluid, especially that of the ions. So these differences, they are not only maintained, but they are actually caused in the first place by the transport mechanisms which are all the time happening across the cell membranes. Now here on the screen, you can see the classification of these transport mechanisms. And you can see that one of the mechanism, that is diffusion, the other is osmosis, and then comes the active transport. And keep in mind that the rest of the mechanisms which are mentioned here, the endocytosis, exocytosis, and transcytosis, they all are active transport mechanisms, which we will study that the active transport is the mechanisms which need an extra source of energy. So keep in mind that the endocytosis, exocytosis, and transcytosis, they are the subtypes of active transport. But we will not go into the detail of these endo, exo, and transcytosis. In fact, we will look at the mechanics of the energy utilization in the basic processes like diffusion and active transport. And then we will look at the transport of water molecules specifically, which is named as osmosis. Uh, Now you must be wondering that if the endocytosis, exocytosis, and transcytosis, they are the subtypes of active transport, then what is the basic classification of these mechanisms? You see, the transport through the cell membrane, that occurs either directly through the lipid bilayer, or that takes place through the proteins. So we can say that the transport, it occurs by one of the two basic processes, which is diffusion and active transport. Now, the diffusion itself, this could be a simple process or this can be facilitated. While the active transport, we broadly categorize this active transport into primary and secondary active transport. And as I already told you, that the endo, exo, and transcytosis, they are subtypes of the active transport. And in the chapter of cell, I hope that you have studied the endocytosis, that is the influx of the nutrients inside the cell. And that again is of two types, the phagocytosis and the pinocytosis. Phagocytosis is basically the engulfment of the solid particles, while pinocytosis mostly involve the fluid part of the ECF, which is required by the cell. Then transcytosis is a common route for the substances to pass between plasma and the interstitial fluid. So all these basic definitions or the basic concepts, they can make one of your MCQs, the multiple choice questions. So, looking at the basic processes, the diffusion and active transport, what is the difference in these two basic processes? Diffusion actually means random molecular movement of the substances molecule by molecule, either through the intermolecular spaces in the membrane or in combination with a carrier protein. And the energy that causes diffusion, that is the energy of the normal kinetic motion of matter. Now, active transport, it means the movement across the membrane in combination with a carrier protein in such a way that the carrier protein causes the substance to move against an energy gradient, mean in the opposite direction to a difference in the energy concentration. So active transport, it requires an additional source of energy besides the kinetic energy. Here you can see in this figure the basic classification of the transport mechanisms, that either it is diffusion or active transport. Diffusion, of course, that does not need extra source of energy, while the active transport that does require an extra source of energy. Then diffusion that could be simple or facilitated, and diffusion, it can take place through the lipid bilayer, or it can use proteins which are embedded in this bilayer, and these proteins, they could be simply the channel proteins, or they can be carrier proteins. Now, the other form of the transport which uses the carrier protein essentially, that is active transport. But this active transport that requires an additional source of energy. So, my dear, you should be clear that there is a difference in the facilitated diffusion and active transport. Although both they use the carrier proteins, but in case of facilitated diffusion, an extra source of energy is not required. But in case of active transport, of course, this kind of transport always need an extra and a unique source of energy in addition to the presence and the utilization of the carrier proteins. Now, I would tell you the brief basic concepts about these transport mechanisms. First, let's focus on the simple diffusion. See, it is a process by which a gas or a substance in the form of a solution that expands, and the kinetic motion of the matter particles that tries to fill all the available volume. And keep in mind that the magnitude of the amount, the tendency of the diffusing power, that is proportionate to the difference in the concentration. If the concentration difference is greater, then the amount of the diffusing tendency, that also will be greater. Now, the diffusion of ions, which are, the charged particles, This kind of diffusion that is also affected by the electrical charge which these ions are carrying. So whenever there is a difference in the potential, when I say potential, that means voltage. So whenever there is a difference of potential or voltage between two regions, keep in mind that the positive ions, they will move to the negatively charged region, while the negative ions, they will move in the opposite direction. Now there is an important concept of permeability coefficient of diffusion. How do you define it? It is the facility, it is the ease with which a solute it can diffuse through the cell membrane, and of course it depends on certain factors. It depends on not only the nature of the solute but also the nature of membrane. So we want to see that whether the solute that is lipid soluble or water soluble, and the membrane is made up primarily of lipids or the embedded proteins. So all these things they determine the permeability coefficient of diffusion, that how easy or how facilitated the transport mechanism will be. Here is the list of certain factors which affect the rate of the diffusion, that how much diffusion takes place in a unit time. Now, the rate of diffusion, as you can see, that it depend upon certain factors. If we talk about solubility, keep in mind that the rate of diffusion that increases for the lipid soluble substances. It means that the diffusion of lipid soluble substances is comparatively easier through the cell membrane, while the hydrophilicity, which is the attraction toward the water, the attraction of any molecules of any substance towards water. Now, this hydrophilicity that is inversely related to the hydrophilic nature of a substance means that the more a substance is water loving, then its rate of diffusion through the plasma membrane will be decreased, because this factor is inversely related to the lipid loving composition of the plasma membrane. And if the diffusing substance it carries an electrical charge, then the diffusion it becomes slow. And keep in mind that for certain reasons, our cell membranes they favor the anions over cations, and I hope you know the difference between anions and cations. Anions are negatively charged particles, while the cations they are positively charged particles. But whatever the charge is, our cell membrane, it does not favor the passage of charge carrying substances. So whenever the substance it is carrying a charge, no matter positive or negative, the diffusion through the cell membrane is getting slow. Molecular size is also a factor which determines the rate of diffusion. If the size is greater, then there will be lesser diffusion. And the other thing which our cell membranes they do not easily allow to pass, these are the polar molecules. So if the diffusing substance it has polar molecules, then the permeability, the ease with which they pass through the cell membrane, that will be comparatively small. Now, simple diffusion, it can occur through the cell membrane by two pathways. The diffusion of lipid soluble substances that occurs through the lipid bilayer, while the diffusion of water and other lipid insoluble molecules that takes place through the protein channels. So let's see that how the diffusion that takes place through the protein channels, and we call these protein channels, sometimes we call them pores also. These pores, they are composed of integral cell membrane proteins that form open tubes through the membrane, and they are most of the time they are always open. uh, some of these protein pores, they are called aquaporins, which are the water channels, and they permit rapid passage of water through the cell membranes. But at the same time, they exclude other molecules. They allow only and only the water molecules to pass through the cell membrane, and these kind of protein channels, they are called aquaporins. Now, do these protein channels, they allow every kind of substance to pass through? No, because these protein channels, they have certain characteristics which determine that which substance is going to pass through the membrane and which is going to be hindered. So, the characteristics which define the permeability of these protein channels is their selective permeability, and then the gating mechanism of these protein channels, and this gating or the control of the flow provided by the protein channels. This could be a voltage gating, or it could be a chemical gating. So, when I say the selective permeability, what do I mean by that? It means that these channels, they are permeable for certain substances, and at the same time they are impermeable to certain other substances. And this selective permeability, that depends upon the characteristics of the channel itself, such as its diameter and its shape. And it also depends upon the nature of the electrical charges which are present inside the channel, and also on the chemical bonds which are formed between the molecules of the channel protein itself, and also the chemical bonds which are formed between the transporting substance and the molecules of the channel protein. Here you can see in this figure that is a protein leak channel, and leak channels, they are always open, but they also have a specific permeability, because every leak channel, even that leak channel has got a selectivity filter. It means it is selective in transporting one or two kind of these substances. Now, this potassium channel is specific for the transport of potassium ion only. And how does it become specific for the potassium ion only? That depend on the diameter of the channel, then its shape, and then the chemical bonds and the charges which are lying inside the channel. So all these factors, they determine that the ion which is going to pass through this channel, that will be potassium only. Now, the second factor which determines the transporting substance, that is the gating of the protein channels to control the transport specifically, and then the rate of the transport. Gating of the protein channels that provides a means of controlling the ion permeability of the channels. And the gate like extensions, they can close the channel or they can be lifted to open the channel. Now, in case of voltage gating, the molecular conformation, the molecular shape of the gate, or the shape, the molecular conformation of its chemical bonds, that responds to the electrical potential, the electrical difference across the cell membrane. In this figure, you can see that when there is a strong negative charge on the inside of the cell membrane, this causes the outside sodium gate to remain tightly closed. And when the inside of the membrane, this area, the inside of the membrane, it loses its negative charge, then these gates, they open suddenly and they allow the sodium to pass in. And I hope you already know that in the ECF, the concentration of sodium ions is way greater than that in the inside of the cell. So, to control the concentration difference and to control the transport of these sodium ions through the gated channels, these gates, they are voltage controlled. As long as the voltage inside the membrane that remains negative, then the gates of the sodium channels, they remain closed and they do not allow the entry of sodium inside the cell. But as soon as the negativity inside the membrane that becomes less or it changes in the positive direction, these gates, they open and they allow a rapid influx of the sodium ions inside the cell. Now there is another example of voltage gating. Here you can see that the potassium gates, they are on the intracellular end of the potassium channel, and they also open when the inside of the cell membrane that becomes positively charged. And when they open, they allow the outflux of the potassium ion to the outside. So, as long as the membrane, the inside of the membrane that remains negative, the gate which is located on the inside of the potassium channel, that will remain closed. But as soon as the negativity it decreases or it goes towards the positive direction, the inside gate of the potassium channel, it will open and that will allow the transport of potassium ion from the ICF to the ECF. Right. So I hope that if you look at the clinical aspects of this voltage gating, then you might have some interest in this mechanism. There are certain disorders which are linked with the malfunction of these voltage gated channels. There is an example of night blindness, which is, you see, it's a recessive disease of the human retina, in which the mutation that takes place in the voltage gated calcium channel, and that mutation that results into a defective a neurotransmitter release, and a defective neurotransmission. And so the patients, they have difficulty adapting to the low light situations, and they also have reduced visual acuity. They might have myopia and other symptoms. Another example of the malfunction because of the mutation in the voltage gated channels, that is the Lambert-Eaton myasthenic syndrome. Now, this is an autoimmune disease which is characterized by the antibodies which are formed against the voltage gated calcium channels, and that again leads to defective neurotransmission and the weakness of the proximal muscles. So, you see that how important the control of the channels that is. And this control is achieved through voltage gating or the chemical gating. You can see that how a difference of the voltage across a cell membrane that controls the opening and the closing of these gate-like extensions and hence the rate of the diffusion. Another type of the gating to control the rate of the diffusion, that is the chemical gating. You see, some protein channel gates, they are opened by the binding of a chemical substance with the protein, and that chemical substance in that case, it is called a ligand. Now, this binding, it causes a conformational or the chemical bonding change in the protein molecule that opens or closes the gate. An example of a chemical gated channel, that is acetylcholine channel. This acetylcholine, actually, this is the ligand which opens the gate of this channel. This acetylcholine gate, that is important for the transmission of the nerve signals from one nerve cell to another or from the nerve cells to the muscle cells to cause muscle contraction. Um, to spark your interest, I give you the example of the malfunction of chemical gating. You see, there is a disorder which is called myasthenia gravis, and this is an indirect ion channel disease which is produced by an autoimmune disorders. What happens that the autoantibodies, they are formed against these acetylcholine receptor. So when they are formed against the receptor, they destroy the receptor, and there is lower concentration of the receptor. And now, this decreased number of the receptor, that results in the blockage of neuromuscular transmission, and the individuals with this disease, they experience again the weakness of skeletal muscles. I hope you can understand that how important the gating of the channels is in the control of the rate of diffusion, and the gating could be voltage gating or the chemical gating. Voltage gating is operated by a change in the voltage across the membrane, while the chemical gating, that is controlled by the attachment or the bonding of a chemical substance with the channel. And in this case, the chemical, that is called a ligand. Now we will move on to the facilitated diffusion, which is again a passive form of transport across the plasma membrane, in which the substances, they move down their concentration gradient which means that they move from the higher concentration to the lower concentration, and they do not consume extra source of energy during the facilitated diffusion. However, they need the help of a carrier, and there are specific integral protein which serve as water-filled carriers. Now, the rate of the facilitated diffusion that is determined by the size of the concentration difference, the concentration gradient, and that also depend upon the number of the carriers which are available at that time for the facilitated diffusion. And keep in mind that these carriers, they are not random carriers, they also demonstrate specificity, that the carrier is specific for the substance which it is transporting. Here you can see the list of the substances which prefer the mode of facilitated diffusion. And these include mostly ions, urea, which is a waste product, then the nutrients such as glucose, fructose, galactose, and certain vitamins. A facilitated diffusion that is also called carrier mediated diffusion because a substance that is transported using a specific carrier protein, and during which the carrier it facilitates the diffusion of the substance to the other side. So here you can see that how the carrier that is helping the facilitated diffusion, and the carrier at the same time it is showing a specificity also. It has specific receptors for the binding of the substance which has to be transported, and then the carrier it undergoes a change in its shape and it transports the substance from one side of the membrane to the other side of the membrane. There is an important concept regarding the facilitated diffusion, and that is called VMAX. This VMAX is basically the maximum rate a facilitated diffusion can attain. In the facilitated diffusion, the rate of the diffusion it approaches a maximum, and that maximum is called Vmax. Here the V stands for velocity and max is of course maximum. So it reaches the rate of the facilitated diffusion, it reaches a Vmax as the concentration of the diffusing substance increases. You can see the difference in simple diffusion and the facilitated diffusion, that as the concentration difference of the substance that increases, our simple diffusion it keeps on increasing. The rate will keep on increasing in case of simple diffusion when the difference of the concentration across the plasma membrane that keep on increasing. But in case of facilitated diffusion, there comes a ceiling, and the rate of the facilitated diffusion it cannot reach beyond this ceiling. Now this ceiling that is called V max. This V max is the maximum rate of the facilitated diffusion which a substance can gain, even though the concentration gradient of the substance that keep on increasing. But the facilitated diffusion it will not go beyond the Vmax, while the simple diffusion it can increase indefinitely in case of an increasing concentration gradient. Among the most important substances that cross the cell membranes by the facilitated diffusion, these are the glucose and most of the amino acids. Well, now here comes your university question, and that's very important. That how can we compare these two types of the diffusion, the facilitated and the simple diffusion, how they are similar and how they are different? You see, both type of diffusion, they take place along the concentration gradient, means from the higher concentration to the lower concentration. But no carrier protein is involved in the simple diffusion. And then the rate of the simple diffusion that can also be influenced by a change in the temperature and pressure. But our facilitated diffusion, it rarely depend on the temperature change. Now, as I already told you that the simple diffusion, it increases indefinitely with the concentration, with the increasing concentration gradient. But our facilitated diffusion, it cannot go beyond the Vmax. There is a limit to an increase in the rate of facilitated diffusion, because this facilitated diffusion, that is depending on the number of the carrier proteins which are available at that particular time. Our lipid soluble substances, they prefer the route of simple diffusion, while lipid insoluble substances, they follow the route of facilitated diffusion. And keep in mind that the uh, carriers, the protein carriers which are used in the facilitated diffusion, they are not random carriers. These are specific carriers. So we have to look at the factors which affect the net rate of diffusion. So one of them is the concentration difference. You see, the net diffusion rate, it is directly proportional to the concentration difference across a membrane. Generally, whenever the concentration difference across two sides of the membrane, when this difference increases, the rate of the net diffusion, that also increases. And how do we determine this net diffusion rate? For that, we use an equation which shows that the rate of the net diffusion, that is directly proportional to the concentration of the substance outside the membrane and inside the membrane. The next factor which affect the net rate of diffusion is the effect of membrane's electrical potential, which is called Nernst potential. And this effect is evident on the diffusion of ions. Now, how do we define the Nernst potential? Uh, it is basically an electrical difference across a cell membrane which balances a given concentration difference. This is, of course, a difficult concept, but let me explain this to you. You see that there is a concentration difference of the charged particles. Um, so, if we consider this as the outside and this is the inside, or simply the side 1 and the side B, there is a greater concentration of the negative particles on one side as compared to the other side. So, the diffusion, it will start taking place. Since the particles, they are charged, there will come a time when there is enough number of the negatively charged particles on the other side of the membrane that they will not allow further movement of the negative charges toward this side, because there is already too much negativity, and this too much negativity is going to repel any further negative charges. So here at this point, the movement of the negative charges that will stop, and this change in the voltage across the membrane, which has resulted from the transport of some amount of the negative charges on the other side, that is called Nernst potential. Because at this Nernst potential, these negative charges, they have stopped the further movement of the negative charges to this side, although there still exists a concentration gradient across both sides. This concentration gradient, it can drive more negative charges towards this side, but the negativity which is already present now, which is building up on this side, this negativity is going to stop these negative charges. No matter how much concentration gradient is present of the negative charges on both sides. So you can say that the difference of the voltage, which cancels the effect of the concentration difference, that is called a Nernst potential of the membrane for that particular ion. And our membrane, it has Nernst potential for almost all the charged particles, and each charged particle, it has its own specific Nernst potential. So how do we calculate this Nernst potential? For that, we use this Nernst equation, in which this EMF, it stands for the electromotive force between side 1 and side 2. And keep in mind that the Nernst potential, that is maintained at the normal body temperature of 37 degree centigrades. Mean there is always a Nernst potential for each ion, and there is always a Nernst potential for each cell membrane at which the movement of the charged particles, it is stopped at a particular concentration difference across the cell membrane. And that difference in the voltage, that is called Nernst potential. Now, the third effect, which controls the net rate of diffusion, is the effect of the pressure difference across the cell membrane. And the example of this effect, that is the pressure development at the capillary membrane. The pressure difference in this case is around 20 millimeter of mercury greater inside the capillary than the outside of capillary. And this results in increased diffusion from the high pressure to the low pressure. So, if I summarize the main factors on which the net diffusion depends, these will be the concentration difference, then the voltage difference, and finally the pressure difference. Now, I will just briefly tell you there are some special types of passive transport, and they are listed here: bulk flow, filtration, and osmosis. Bulk flow is the diffusion of large quantities of substances from a region of high pressure to the region of low pressure. And this bulk flow is due to the pressure gradient across the cell membrane. For example, I give you the instance of the exchange of gases across the respiratory membrane in the lungs. You see, the partial pressure of the oxygen that is greater in the alveolar air than in the alveolar capillary blood. So, oxygen it moves from the alveolar air into the blood through the respiratory membrane. Similarly, the partial pressure of carbon dioxide it is more in blood than in the alveoli. So, this CO2 it moves from the blood into the alveoli through the same respiratory membrane. Well, the other special type of passive transport is filtration. The movement of water and solutes from an area of high hydrostatic pressure to an area of low hydrostatic pressure, that is called filtration. I repeat again, the movement of water and solutes, not only water, the movement of water and solutes from an area of high hydrostatic pressure to low hydrostatic pressure, that is called filtration. And the hydrostatic pressure itself, that is developed by the weight of the fluid. This filtration process is seen at the arterial end of the capillaries, where the movement of fluid it occurs along with the dissolved substances from the blood into the interstitial fluid. And then the filtration it also occurs in the glomeruli of kidney. Well, the diffusion of water molecules across a selectively permeable membrane, it gets a special name, and that name is osmosis. Well, before getting a grip on osmosis, the process of osmosis, one has to understand uh, what is a selectively permeable membrane in this process. You see, a membrane which allows water but not the solutes dissolved in water to diffuse through the membrane, that is called a selective membrane for the osmosis. Now, why do we have to understand the process of osmosis? Because water is the most abundant substance which diffuses through the cell membrane. A concentration difference of water it can develop across a membrane, and it acts as a driving force for osmosis. And when this happens, net movement of water does occur across the cell membrane. So by definition, the process of net movement of water caused by a concentration difference of water, that is called osmosis. Direction of the osmosis is from less concentrated solution to more concentrated solution. Now here the concentration of the solution that is described in terms of solute concentration and not the water concentration. So what is osmotic pressure? The exact amount of the pressure which is required to stop osmosis, that is called osmotic pressure. And that is when the concentration of the solutions on either side of the membrane that becomes equal. Uh, so, which factor determines the osmotic pressure? This determining factor is the molar concentration of the solution. Well, the molar concentration of the solution basically that is the number of the particles per unit volume of the fluid, because these particles, they are osmotically active. The concentration of these osmotically active particles, that is usually expressed in osmoles. Now, you must be wondering that what is osmolarity and osmolality? You see, the osmolarity, that is the number of the osmoles per liter of the solution, whereas the osmolality, that is the number of osmoles per kilogram of solvent. This concept is important. The term tonicity, that is used to describe the osmolality of a solution relative to plasma. The solutions with the same osmolality as plasma, they are said to be isotonic, while the solutions with greater osmolality than plasma, they are called hypertonic, and similarly the solutions with lesser osmolality than plasma, they are said to be hypotonic. And here in this figure, you can see that how does the tonicity of the fluid, it can affect our body cells. So if the solution is hypotonic, then the movement of the water, that will start from outside into the cell, that will increase the cell size, and that can also result in the bursting of cell. And if the solution that is hypertonic, means its osmolality is greater than that of plasma, then the water present inside the cell, that will start diffusing from inside to the outside, and cell size will shrink, and this is called the cell crenation. All right. So, what is an isoosmotic solution? You see, these solutions with the same concentration of the solute particles, they have same osmotic pressure, so they are called isoosmotic solutions. The solutions, they are still isosmotic even if the type of solutes, they are different in two solutions. Well, the types of the osmosis which occur across the cell membrane, that is um, endosmosis and exosmosis. In endosmosis, the movement uh, of the water that takes place into the cell, while in exosmosis, the movement of the water that takes place out of the cell. Right, so what is reverse osmotic pressure? You see, it's a process in which the water or other solvent, it flows in reverse direction, means from the area of higher concentration to the area of lower concentration of the solute. In case the external pressure that is applied on the area of higher concentration. So the reverse osmotic pressure, it occurs or it develops only when an external pressure is applied on the area of higher concentration. Okay, the development of colloid osmotic pressure, or we can call it oncotic pressure, you see, it's a process exerted by the colloidal substances in the body, that is called colloidal osmotic pressure. And the osmotic pressure that is exerted by the colloidal substances, such as the proteins of the plasma, that is known as the oncotic pressure. And its value is about 25 millimeters of mercury. Now, this figure is basically demonstrating the movement between the fluid compartments during dehydration, and this is how the osmosis it works in restricting the water movement, because our body it puts mechanisms in place that independently control the water content, and that's how the net water movement that is limited between the intracellular fluid, extracellular fluid, and plasma. That you can see that if the person is excessively sweating, then how do our kidneys they help in maintaining the homeostasis, the water homeostasis of our bodies? Uh, our blood it contains large amounts of albumin, around 3.5 grams to 5 grams per deciliter of the plasma. Now, this albumin that is trapped in the vascular compartment by virtue of its large size, and its function is to help create an osmotic potential which draws the extracellular fluid into the vasculature. Now, keep in mind that this osmotic potential that is known as plasma colloid osmotic pressure.